My name is Paul Lombroso. I'm from the Child Studies Center, and today I wanted to follow up a little bit on some things that I introduced yesterday in a lecture on learning and memory. What I'm going to be talking about is some observations that Flora alluded to very briefly in one of her slides, is that children who have Tourette syndrome often have a number of comorbid disorders, and about half of them have obsessive compulsive disorder, and about half of them have attention deficit disorder. A typical child that we'll see in our clinic, or about 10 to 15% of the children, have a history of being seen first, before they even come to our Tourette's movement disorder clinic, of having a history of attentional problems, being seen by a pediatrician, being placed on a stimulant medication, and then coming down with a movement disorder, and then they come in to see us. My lecture today, what I wanted to do is start from that clinical observation and tell you a little bit about how we move from that clinical observation into the laboratory. So it's been known for many years that in fact you can develop an animal model of a movement disorder using amphetamine. And so you can give, for example, daily injections of amphetamine, say for 5 days, then give the animals, in this case we're talking about rats, give them a week off and challenge them then at about 10 days later with a very low dose of amphetamine. And these animals will then have what we call a stereotypic repetitive movement disorder that will develop. I want you to see it. This is a normal rat, has been injected for 5 days with saline. Okay? Nothing unusual, just this is the animal that's been injected with amphetamine. And we'll spend the next hour or so in this repetitive nose-down movement. Okay? I'm not saying that this is a tic disorder, but what I am saying is that it is a movement disorder that we can now bring into the laboratory and ask some questions. This has a long history of being studied by many investigators throughout the world. My interest was in trying to determine whether or not we could block the induction of these movements. And I'll show you how we do that. So yesterday, one of the main points of the lecture was a discussion about synaptic plasticity, and I'm going to just mention this briefly today, repeat some of this. One of the things that happens during the development of synaptic plasticity, as spines change their shapes, new spines are made or old ones are strengthened. And about 10 years ago, an investigator by the name of Terry Robinson decided to test whether or not there was a change in the number of spines after repetitive amphetamine injections. Saline injection is here. I'm sorry, you're not getting a good sense of this. The next slide, these are the amphetamine injected animals. And we're going to look now at a much higher powered look at spines that he examined. So here's the saline injected spines. And you can see, I think you can appreciate that there are quite a few more, there are many more on the amphetamine. This is taken from the nucleus accumbens. This particular is from the core of the nucleus accumbens, but similar results were obtained from the shell of the nucleus accumbens. Over here you can see the change in the number of spines. There was also a change in their complexity. So rather than, sometimes spines have different bifurcations, and these are called more complex spines. And there was also a very significant increase in the complexity of the spines that were formed. Now, there is a protein that I introduced also yesterday during my lecture. It's called ERK. It's required for these structural modifications. As you can imagine, the changing in the shape and the number, the density of these spines is going to require de novo protein synthesis. And this protein ERK is involved in that process. A summary slide that just shows you what we're talking about. There's a new model about how the proteins that are delivered to these spines, how they actually arrive here at the spine heads. The old model suggested that, as you know, the mRNA, or the messages that are encode for these proteins, are delivered into the cytoplasm, where they're translated into proteins. The new model suggests that actually the messages themselves are delivered, out to the spines, and they sit there quietly, waiting for some type of synaptic input to arrive. When a particular spine is in fact activated by synaptic activity, those messages are present there and are then translated right there at the spine and delivered to the spine for the morphological changes that then develop. So the current model says that messages, yes, indeed, they're produced in the nucleus. They're brought out by actually one of the proteins that is known to help to transport these messages is a protein that is mutated in Fragile X syndrome. And if people are interested, we can talk more about that at another time. But proteins escort these messages out along the dendrites. They are delivered, as I said, to all the spines, and they're sitting there quietly waiting for a signal to arrive. And when that signal, in this case large activity, synaptic activity through the presynaptic neuron, that activates this protein that I mentioned. That activates, the messages are then translated, the proteins are then produced locally, and inserted into the spine. This is a completely new model that has now a tremendous amount of data that supports this model. The delivery of the messages directly to the spines, they are inhibited from being translated, a signal arrives, and they're locally translated. This takes care of a problem that people had in the old way of thinking of things, was how did the proteins know where to go? I mean, we have 10,000 spines, and we want synaptic plasticity to occur at 10, 15 of them. How do those proteins that are synthesized here know which spines to go to? And people had complicated models. This model is much more sophisticated. It's a much simpler model that explains this particular problem. But I did say that there was a protein called ERK. ERK, in fact, one of its roles is to activate the translation, to trigger the translation from these messages into the proteins that are needed for the morphological changes that are occurring at the spines. One of its functions, as I said, is to regulate. It's one of the proteins that regulates local protein translation. Another function of ERK is to rapidly move into the nucleus. And what it does in the nucleus is initiates transcription of new messages that are then in turn transported down. So ERK has two functions. It has more than two, but two I'm discussing today is the regulation of the local translation and the initiation of transcription of new messages. I thought you might actually be interested in seeing phospho-ERK, p-ERK. The experiment here is using primary cell cultures, and there are two antibodies that are being used to look at these cell cultures. One recognizes ERK, which is not phosphorylated. So what we call this a total ERK. And you can see that in this slide, it's green. The other antibody that is used only recognizes ERK when it has been phosphorylated, i.e. activated. And here we're stimulating these cells with glutamate, At time 0, we have lots of ERK, but it's not yet been activated. Within a very few minutes, 5 minutes in this case, it has in fact been activated. And in fact, if you look carefully, you can see that the large majority of this ERK is where? It's been translocated into the nucleus, and it's beginning to initiate the transcription of new messages that this signal, this stimulation, initiated. And within a few minutes, it disappears. But its job has already been done. If one could look, there would be messages being transcribed in the nucleus. Okay, so this is p-ERK. And it's a key player in synaptic plasticity. If you cannot activate ERK, if you cannot phosphorylate ERK, you cannot, synaptic plasticity does not proceed. And that is what is required for consolidation of memories, as we discussed yesterday, and for the sensitization to repetitive amphetamine injections. In all parts of the brain, as I showed yesterday, in all parts of the brain where memory is occurring, whether it's declarative or non-declarative, p-ERK is required. It has to be activated, and in all parts of the brain where people have disrupted the activation of ERK, they've disrupted the specific memory that occurs in that part of the brain. What regulates ERK? There's a protein that my laboratory studies. It's called STEP. It stands for striatal enriched tyrosine phosphatase. What that means is that it regulates ERK, and it regulates ERK by removing a phosphate group. So when a signal arrives, either a growth factor or synaptic activity, it leads to this rapid activation of ERK through a series of kinases. Kinases add phosphate groups, and so RAF is a kinase, it activates MEK, and MEK in turn is a kinase, and it phosphorylates and activates ERK. You have to carefully regulate this process, and this particular protein, STEP, is one of the players that regulates by dephosphorylating, removing the phosphate groups from ERK, and thereby inactivating it. STEP is expressed in this slide. I'm showing where STEP is expressed. In fact, it's expressed highly in the striatum, that Flora spent so much time talking about. It actually, more recent work has shown that STEP is expressed in all parts of the brain where learning occurs. It's present in the hippocampus, in the nucleus accumbens, in the striatum, in other parts, and in the cortex itself. We're going to look a little bit more at higher power here, and you can see this is STEP immunoreactivity in a neuron, and it's expressed throughout the neuron. Okay. I mentioned that STEP regulates ERK's activity, and that's just shown here. You can see that it dephosphorylates this particular amino acid. It's called tyrosine that gets phosphorylated. If ERK is not phosphorylated here, it becomes inactive and can no longer signal. Okay? So that's the background on ERK. So here's the question that we wanted to test. Could we make STEP inactive, infuse that inactive STEP into the relevant brain region, in this case it would be the striatum, and block amphetamine sensitization and the development of stereotypies? This is how we do it. I'll spend just a second explaining this. So we have STEP in the lab. I cloned it years ago, and so we can actually grow it up in bacteria. We can put, in addition, what's called a point mutation into this particular the message that encodes for STEP. A point mutation that switches a particular amino acid. The useful active STEP has a cysteine, and we make a single nucleotide change. Cysteine is no longer placed, but instead a serine. The effect of that single mutation is that STEP itself is now inactive. We do a couple of other tricks. We add this particular sequence here. It's a sequence of about 13 amino acids. It's called TAT. And the only reason we do that is that this has the ability of burrowing its way into cell and pulling the protein that you have of interest into those particular neurons or into those particular cells. So this is a way of getting the inactive step into the cells that we want to study. Now, so we've made this point mutation. ERK still binds to STEP, but STEP is no longer able to dephosphorylate ERK, and because of that, ERK is trapped. It's not released, and a complex is formed that consists of ERK and STEP. So in that situation, we hypothesized that perhaps this key signaling protein, ERK, would no longer be able to do its role. It would no longer initiate local translation. It would no longer initiate transcription within the nucleus, and perhaps we could block the synaptic plasticity that occurs. Okay? Now we have to get it into the brain. I'm just showing you here what I just said, that we were hoping to be able to bind to ERK with this step and block either local translation and or transcription of new messages, and therefore block the development of synaptic plasticity. Here's the experimental design. We perform surgery. In this case, we're placing cannulas bilaterally into a particular region of the striatum called the ventrolateral striatum. The ventrolateral striatum has been known for a long time to be a key regulator of amphetamine-induced stereotypies. So we decided to try to infuse our STEP mutated protein into the VLS, ventrolateral striatum. After the surgery, placing the cannulas into the VLS, we wait a week for the animals to recover, and then we infuse our mutated STEP protein. We wait about an hour or so, allowing the STEP protein to then get into cells. We could actually check that, in fact, if we did sections through this part of the brain actually within a few minutes to several hours, up to 10 hours afterwards, the STEP protein that we had infused was present inside neurons. Each day, we then give an injection of amphetamine about 1 hour after our infusion of STEP. We do this for 5 days. So the next morning, we infuse the STEP and inject the amphetamine, and do that for 5 days, and then wait for about 10 days and challenge with a low dose of amphetamine. So this is just showing you actually that when we actually look, we can find that this in green is the STEP mutated protein, and it's within these neurons here. This was actually done in cell cultures, but we also did it in the ventrolateral striatum. Okay, so this is now a control animal infused with just a control peptide at day 10, and spends the next hour or so with this. In this case, it's not a head down, it's a head up repetitive stereotypy. And here are the experimental animals. It appears that we've blocked the induction of these stereotypies. Okay, we could quantify it. We've done this. So these are the control animals, and we just simply are counting here the number of up and down head sniffs, and there's a highly significant decrease in the number of these stereotypies. Only though if you inject the STEP during those 5 days when sensitization is occurring. So you can imagine what happens if you inject, so you give your amphetamine for 5 days, but now you inject the STEP, or you infuse rather the STEP at a later point, after those 5 days. That's shown here. There's absolutely no difference then. Okay? So the infusion of STEP has to be done during the 5 days when sensitization is developing. So this is my last slide. I just want to end here. What I was hoping to share with you is one experiment, one way of looking at translational neuroscience, taking as a starting point the fact that many children who have Tourette's syndrome, their tics can be worsened or even initiated by amphetamine, taking that observation, that clinical observation, and then looking at an animal model to see if we can find ways of disrupting the induction of stereotypies. And we used the fact that STEP, the protein that our lab works on, regulates several actual proteins, signaling proteins that are critical during the development of synaptic plasticity. Today we only had time to talk about ERK, but STEP also regulates a critical glutamate receptor called an NMDA receptor. It dephosphorylates particular subunits on these receptors, as well as another key signaling protein called FIN. And because these proteins are required for the development of synaptic plasticity, we wanted to test whether in fact we could block the amphetamine-induced stereotypies, and in fact we were able to do that. Thank you very much. Let me just thank, of course, the people who have done this work and my various collaborators.